Hello everyone, I am Puneet Paul, working as a postdoctoral research associate at Department of Agronomy and Horticulture here at University of Nebraska-Lincoln. Um, First, I would like to thank the organizers for giving me the opportunity to present my work. And today I'll talk about some of the highlights of my work that focuses on the physiological and molecular perspectives on reproductive development under high temperatures. And please do not hesitate to contact me back for any questions. And my email account is puneet6288 at gmail.com, or you can also contact me on Twitter. And my handle name is Paul Puneet. And so let's begin. And I would like to minimize the video here. Um, so other than the ongoing pandemic, we as a society are facing massive challenge in the name of global climate change. And this changing climate has already led to a rapid increase in temperatures over the last century or so, as you can see from this line graph. And this increasing temperatures are highly correlated with the decline in the yields, decline the rate of yields for major crops such as wheat, rice, and maize. And now, when you bring in the ever-growing population into the picture, the situation becomes deeply concerning, as this scenario has caused a number of issues, and one of them is food insecurity that not only leads to hunger, that means lack of food, but it also leads to hidden hunger, that means lack of food with proper nutritional values. And one solution to avoid or to overcome this problem is to create climate resilient crops that can withstand high temperatures. So whenever we talk about resilience to heat stress in crops, it's always the reproductive development that comes in focus. And reproductive development here, I mean, is that encompasses both the pre and the post-fertilization events. For instance, whenever the plants are subjected to heat stress during pre-fertilization, it's usually the pollen that is very sensitive, and as it leads to non-viability as well as abortion of pollen tubes, that in turn leads to reduction in yields due to higher temperatures. And whenever the plants are subjected to heat stress during post-fertilization, it's usually the developing grain that's very sensitive. And heat stress basically causes reduction in the sink capacity, decreases the grain size and or weight, which in turn causes the eventual loss in yield. So for my PhD work, I focus on the pollen side under heat stress, and my postdoc work was a natural extension of my PhD work, where I'm focusing on the post-fertilization events, how developing grain cope up with the higher temperatures. So today I'll focus only on the post-fertilization, and going back in 2016 when I joined Harkamal's lab here at University of Nebraska-Lincoln. It was also the time when he was funded for a huge grant to develop or create this wheat and rice center for heat resilience in partnership with Arkansas State University as well as Kansas State University. So the main objective of this program was to develop or discover genes underlying heat stress resilience in cereals, mainly rice and wheat. And my role in this program was to lead the studies on rice side, where we wanted to understand how high night temperatures impact rice, and also to co-mentor graduate students under Harkamal supervision. So So you might be wondering that why we only focus on high night temperature stress. So to understand this, we have to look at long-term climatic records. And for this, we collaborated with an excellent scientist, Meet Palkukal, who works across the street in Department of Biosystems Engineering here at University of Nebraska-Lincoln, who basically mapped the changes in the temperatures for daytime and nighttime separately for the last century or so. What you are seeing here on the screen is on the left side, how the temperatures have changed over the last century only during the daytime. And on the right side of the screen, you are seeing how the temperatures have been changing for the uh, uh, exclusively for the nighttime over the last century. And if you can see that the increase in the nighttime temperature has been higher or more than the increase in the daytime temperature over the last century. So it's a very big problem that we are looking at. Why so? Because higher night temperature leads to higher respiration in plants, which in turn causes losses in carbon and eventually causing yield declines. And for rice, where I'm focusing on, There have been a well-established study that claims that with every degree increase in the nighttime temperature, there is at the best there is like 10% loss in the yields for rice. So this basically our study as well as the other studies found made a very good foundation for us to start our experiments. So so I began planning the experiment with yet another excellent graduate student, Balpreet, to understand grain development at a higher night temperature. And if some of you may know that as soon as rice undergoes fertilization, the rice flower opens for a very brief period of time, if you can see here. And we took advantage of this beautiful phenomena, and we marked these open florets to track the seed development later on. So what we did was that as soon as we marked the seeds, we put the plants, half of the plants in control conditions, and the rest undergo terminal high night temperature treatment to assess how high night temperature impacts the grain development. And for this pilot study, we basically focus on six accessions or genotypes coming from different subpopulations in rice, like indica, temperate japonica, tropical japonica. These are different subpopulations that have different genetic backgrounds. So what you're looking here is that under high night temperature, there was only one of the genotype, or we call tropical japonica number 2, that had significant decline in the single grain weight. So this is one instance, and then we also looked at the mature seeds. So what we found was that one of the accession or genotype indica 1 had these black spots on the mature seeds that were subjected to high night temperature, whereas other genotypes, for example, tropical japonica number 3, the seeds in the high night temperature treated plants were as good as under control conditions. So what this black spot is, basically this is called chalkiness, and this chalkiness is mainly because under high net temperature, what happens is that the starch granules, which is basically what we eat from rice or starch, they undergo structural disorganization. And what happens is that under control, the starch granules are polygonal in shape and are very well packed. But what happens in under high night temperature stress, that the starch granules form rounded structures, and these rounded structures are like a room full of balloons where there's a lot of empty space, and this empty space basically captures the light and gives the rice the black phenotype what you're seeing here for indica 1. So why I am making such a big deal about this black spots is because chalkiness is a huge issue in the rice biology, as it leads to millions and millions of losses in dollars for the rice world and the farmers. So what we conclude from this pilot experiment was that high net temperature impacts not only the grain quantity, as you can see from the single grain weight, but it also impacts grain quality and leads to a lot of losses. So learning from this preliminary experiment, then I planned another large-scale experiment where we included 300 different genotypes from rice, which we call Rice Diversity Panel 1. And since it was a massive experiment, so we included another very talented graduate student, Jasprit and Larissa, in this experiment. And these 300 varieties, like I said earlier, not only come from different genetic backgrounds such as indicas, tropical japonicas, or temperate japonicas, but also come from different geographical locations. As you can see, some have originated in India, some have originated in Africa, some have originated in South America or so. So again, we subjected these to high night temperature treatment, and then we phenotype them or screen them using two different methodologies. So one non-destructive phenotyping that I'll talk later, but first I would like to take on the destructive phenotyping. So as soon as the plants mature, we take the panicles or seeds, we dehusk the seeds, like remove the hull from the seeds, and then take the weight of the seeds. And once the weight is taken, we scan the seeds, and for this, once the seeds are scanned, we use those images and feed them in an in-house software seed extractor. What this software does is that it basically gives us different parameters of seed as an output, like we got what's the area of the scanned seeds, what's the length, what's the width, or what's the perimeter. So once we had these parameters such as grain weight and other morphometric stuff, so we use this to map or associate to the genetic markers, which was later on used for gene discovery. And this work was basically done by Wasim, the mapping, who was formerly who formerly was a postdoc in our lab. Once we had this mapping done, so then we looked at which genes are we looking at. So well, from this Manhattan plots, as if you may know that these peaks means some of the polymorphism that lie in the rice genome. And what we found was that there are some known genes for grain length or grain width that was our primary focus. So we caught some of the known genes such as GS3 or SW5 that are known to contribute to the grain length or width for rice. So this basically validated our data set. But other than the known, we also found some novel candidate genes that were exclusively coming for the plants that were from the plants that were treated for high night temperature, which I call HNT. As you see that for grain length here, there is a peak at chromosome number 12 that is coming, and for grain width, there were a number of peaks that were exclusively coming under high night temperature. So one such peak co-localized with a known gene, which is called fertilization independent endosperm or FIA1. So this particular gene is a component of a polycomb repressor complex in rice. And this, from previous studies, we know that this particular gene is exclusively expressed during the seed development. As you can see that these bar plots show the expression of this gene, which is only coming in the developing seed at 0 to 11 to 20 days after fertilization. And the expression for this particular gene in vegetative tissue such as root or leaf is almost negligible. So this plus this gene is also responsive to temperature stress. As you can see that when we look at the expression of this gene from developing grain, we saw that the expression of this gene keeps on increasing with every 2-degree increase in temperatures from 28 to 34, and then it starts showing a decline. So all in all, this was an excellent candidate to further characterize for its role in defining grain size under high night temperature, because this particular gene co-localized with a SNP that was exclusively coming in the HNT, and this gene particularly shows seed-specific expression, as I talked here, and it also is responsive to heat stress. So to give you more insight, so this particular SNP that I'm talking about here divided the whole population that we were dealing in two subgroups. So one was called major and the other was called minor, because the majority of the genotypes that we were dealing had the CC allele, so we called them major. Other or very few genotypes or accessions had a GG allele, so we called them minor allele. So what happened was that this particular SNP CC gives resilience to the major genotypes, whereas the minor accessions having GG allele were sensitive to high night temperature. So just to give you an instance, so we look at a few representative of the major group and the minor group. What I am saying here, if you did not understand earlier, what is that the major allelic group that have the CC allele for this particular SNP are not sensitive to high night temperature, because if you see that the mature seeds for this major allelic accession M1, capital M1, are good, as good as under high night temperature as they are under control, and this is true for the 4 representative major allelic accessions. But when we look at the minor allelic accession that have the GG allele, these accessions are highly sensitive to high night temperature, as was seen from the plots. So what you see here is that the plants under high night temperature have a decreased grain width. If you see here in all the accessions, they have decreased grain width. So to further understand why so we looked at the expression of this particular gene. We saw that all the major allelic accession, denoted by capital M, have higher expression for VA1 under high night temperature, wherein the all the minor allelic accession have lower expression for VA1 under high night temperature. So what this means is that if at all we take a major allelic accession and delete this gene, it should start behaving like a minor allelic accession, which is sensitive to high night temperature, as you can see from the grain waves. And yes, that's exactly what we did. So we took a major allelic accession and created a knockout mutant for this particular gene. So what we observed was that when we did CRISPR knockout, so we saw that the major allelic accession having a FIA1 deleted uh, was became highly sensitive to high night temperature with respect to grain width. As you can see, the wild type here has more or less similar similar grain widths under control and high night temperature, as this is not significant. But when we deleted FA1, the grain width becomes highly sensitive to temperature stress. And this was not true only for grain width, but also the grain length, which was compromised in the FA mutants or CRISPR knockout mutants of FA1 in the major allelic accession. So what we did was that we characterized the role of FA1 in grain width regulation under high night temperatures. So so this was about how we went on from conventional phenotyping methodologies, where we looked at the mature seed phenotype at the end of the harvesting. But we also looked at the developing panicle phenotyping, which we call non-destructive, because we did not kill the plants during the process of phenotyping them. And So what we exactly did here was that we built up a platform we call panicle imaging platform, or PyPlat, where we use this to understand the yield related traits at a high spatiotemporal resolution. So what we did was that we phenotyped the plants under control and high night temperature stress at week 1, week 2, and week 3 using this PyPlat method. So here, if you see here the pipeline methodology, we bring in the plant, and the plant goes into this platform, and there are then these two camera systems that then start taking photographs of the developing panicle. And then once these images are taken, then we use all these images to construct the panicle in a 3D manner, and then extract the traits. And here we talk about two traits, basically the voxel count, that basically is the volume of the panicle, and then the color intensity, that means red, green, and blue channel for that we extracted from these images. So basically what we did was that we used conventional phenotyping methodologies to hunt the genes, and then we use this novel phenotyping methodology that we call PyPlat to look at how the plant develops under high night temperature stress. You can have a look at this methodology in our plant method paper that we published recently. So, well, the next question, without going into details of this method, what we asked from this methodology was that can we use this PyPlat for gene discovery? And to answer this, we again looked at the rice diversity panel at different time points like week 1, 2, and 3 after fertilization. And then we screened them using this PyPlat, and then associated this trait, that voxel count and R2 gene intensity, to map on the genetic markers, which was done by Maliki Campbell, a former postdoc in our lab. So what we found was that there are some genes or some peaks that were coming for control, and there are some genes that were exclusively coming under high night temperature. And this is a very complex and a huge data set that we are still trying to make sense of. And what I would like to tell you here about this data is that this basically has opened new avenues to score hidden phenotypes that were not basically looked at from the conventional phenotyping methodology. And this has already helped us in discovering the genes that were not detected from the regular phenotyping methodology. So With this, I would like to move on to the next chapter of my work. Until now, I have talked about the genes that are like FIA1 or GS3 or GW5, if you remember. So these genes basically are preferentially expressed during early seed development. And this early seed development also happens to be the time where endosperm of rice develops exponentially. And to understand why so, so if we look or cut the sections of developing seeds um, at 24, 48, 72, and 96 hours, you see that, uh, and when you look at the sections, you see that as soon as fertilization happens, the endosperms start dividing mitotically in a crazy fashion without forming cell walls. So what you're looking here inside of the seed, developing seed, is that just a mass full of nuclei which does not have cell wall. And as soon as 72 hours, the cell wall starts forming, and by 96 hours, the cell wall's formation is completed. So basically, this particular stage of development of endosperm from after fertilization until cellularization is highly sensitive to heat stress and is one of the major determinant of seed size. So then we ask the question whether how sensitive is this stage, and to answer that, what we looked at a subset of 9 genotypes for how they transition from a syncytial phase to the cellularization phase. Where syncytial, I mean that the mass full of nuclei without cell wall, and cellularization means that the cell wall formation has completely taken place. So under control conditions, all these 9 genotypes reached cellularization stage by 96 hours. But when we imposed heat stress to these 9 genotypes, some of them could only reach stage 3. That means only a few layers of cell wall was formed by 96 hours that were called category 1. And some genotypes could only reach at stage 2, that is formation of RMS, or just this radial microtubule system, by 96 hours actually. So we call them as category 2. And some genotypes were only at stage 1, that is, they were just having free nuclei without any cell wall by 96 hours. So what we found was that there is a component of natural variation that is already there for early seed development and its responsiveness to heat stress. And I recently have screened RDP1 for its response to transient heat stress during early seed development in the same manner as I screened high night temperature stress that I talked earlier. Plus, we also did some transcriptome analysis during this very sensitive stage of seed development. And what we found was that there are 2 genes, MET78 and MET79, that are preferentially expressed during this stage 2 and stage 3. Stage 3 of endosperm development. So both MET 78 and 79 basically corresponds to the family of the genes which is famously known for flower formation. And both MET 78 79, as I talked earlier, are exclusively expressed or preferentially expressed at stage 2 and stage 3, and their expression dies down as soon as the endosperm enters the cellularization phase. And this is the QPCR data that you're looking at, that these genes are expressed maximally at 48 and 72 hours after fertilization. That means 2 or 3 days after fertilization, and their expression is going down at 96 hours, which basically coincide with cellularization. So to understand what's the role of these 2 genes that are just expressed during this very small window of development, we created our expression lines. So as you know that that the endosperm develops completely or cellularizes completely by 96 hours. So when you overexpress these genes that are coming all in stage 2 and stage 3, so what this does is that it pushes the cellularization to a later stage. And at 96 hours, what we observed was that our expression lines are not cellularized at all. So that means the increase in expression because of the overexpression of these genes pushes the cellularization to the later stage. And when we also created a knockout lines, and what happened was the reverse. So when you knock these genes out, Rather than completing cellularization at 96 hours, the cellularization was completed in 84 hours for the knockout mutants. If you see here, that the knockout mutants here for both the genes are already cellularized by 84 hours, whereas wild type plants still have some way to go. And this is already very much visible at 72 hours, where you see that the wild type has just one layer of cell wall formation, whereas the mutants of MAT78 and 29, the knockout mutants, have already developed multiple layers. So you might be wondering why this guy is talking about or making a big deal about this small window of development. Well, what this means for the seed is that when you overexpress MAT78 or 79, it delays the cellularization, which causes larger seeds. And when you knock out these two genes, it causes early cellularization, leading to smaller seeds. So that's the basically consequence of these two genes that are preferentially expressed during early seed development. And then we created double knockouts, and what we found was that neither of the double knockout from our study had any seed wherein the wild type was normal, and as you can see here in the panicles did not bear any seeds. So this means that these are essential for seed development in rice. And currently what I'm doing is I'm trying to understand the role under heat stress. And with this, I would like to end my talk and thank you again to the organizer for giving me the opportunity. And I would like to acknowledge all my mentors and collaborators, Professor Grover from University of Delhi, Professor Schleif from Goethe University, and Wolfram from University of Vienna, and my current postdoc advisor, Professor Harkamal, and all other collaborators. And I would also like to thank the funding agency, Marie Curie, and the NSF funded for Harkamal, and all my mentees that I have worked with over the years, as well as my current collaborators, Mac, Palak, Wasim, and Vipal. Thank you very much.